Hello, I just wanted to give you all a brief overview of the microbiology topic 3.4, and these are all the main points that I'll go through now. So first of all is classification of bacteria, and the most simple form really is to look at the shape. So this is just looking down the microscope and seeing the shape of them. So there's the coccus or cocci, the rod-shaped bacillus, or the spirillum, which is a wiggly sort of spiral shape. And like I say, that's just done by looking down the microscope. That could help if someone's trying to identify bacteria that might be from a swab taken from a person or from a surface or something like that. That would narrow it down to a certain number of bacteria that it's probably likely to be. A lot of bacteria, they will all have a binomial name, like you've done the Linnaeus sort of binomial naming system, usually a Latin name, and a lot of the time it will have this included in it. So it might say bacillus or something, or cocci in the name, like staphylococcus. Another broad way of breaking up bacteria into groups is to classify them as gram-positive or gram-negative. And that would help if you do this test first of all, just to find out if they're gram-positive, gram-negative. And then that would give you a huge way of just saying, it's not this massive group of bacteria, it's got to be from this massive group of bacteria. So at least it does rule some out. And then it also gives the stain for identifying the shape as well. So it would help break it down really so that you get a smaller number of bacteria to consider if you're identifying. So we'll look at the gram-positive first of all. If you were asked what a cell wall of bacteria is made from, you'd say peptidoglycan. So the peptidoglycan is this layer here. And I've drawn it like that because there's a normal cell membrane that the cell would have, and then above that the peptidoglycan on the outside. And you know what it's made from because you need to use your biochemistry knowledge and sort of prefixes and all that kind of thing. Peptido is obviously related to proteins like peptides. So this is made up a lot of amino acids and protein parts. And then glycan indicates sugar components. So these sort of sticky outer bits here, they're sugar components, and there's sugar components in there as well. So the peptidoglycan forms a mesh, and that's the cell wall. It's also sometimes called murin. So in WJC, we use the term peptidoglycan, but if you see it somewhere else labelled murin, then that's what it is. It's just the same thing. Then the gram-negative bacteria, they still have the cell membrane, so that would be around the cell. Then outside of that, they would have peptidoglycan as well. The thickness of the peptidoglycan varies just depending on the type of bacteria, but it would tend to be thinner layer of it in the gram-negative than in the gram-positive. Because in the gram-negative, there's also an outer lipopolysaccharide layer. And you know what lipopolysaccharide is as well, because lipo is fat and polysaccharide is chains of sugars. So there is lipo, there's the fats here, and then the like in a normal cell membrane. You might also have sort of carrier proteins and things in there as well, and then the polysaccharides as well in there and sticking out like they do. So you have gram-positive and gram-negative. And the important thing about this, it does change the way that bacteria reacts with other things and how it reacts to different sort of chemicals and situations. So for example, antibiotics are less effective on gram-negative bacteria because that provides, that lipopolysaccharide layer provides like a waterproof shield around the outside, and it does help protect it from antibiotics. So it does make a difference. And the way we identify it is with the gram technique or the gram stain technique. And that is done with, you can make up your own mnemonic for this one if you want to, but this is crystal violet. Crystal violet's the actual stain, like the purple stain or dye. Iodine then takes that stain and fixes it. So these are 4 different stages. You start off with crystal violet and then you put on the iodine. And what the iodine will do is fix the crystal violet to the peptidoglycan. So by this stage, you've got the peptidoglycan with the crystal violet stain stuck onto it. It would be visible on here at this point. So because it would have a layer of crystal violet on the outside, but it wouldn't have been fixed with the iodine, because that layer is protected by the lipopolysaccharide, you then have alcohol And the job of that alcohol stage is to basically wash away, because the alcohol will break down the fats, won't it? Break away that membrane. The alcohol breaks away that membrane and exposes peptidoglycan. But in breaking it away and washing that layer away, it washes the layer of crystal violet along with it. So the crystal violet was fixed to this one with the iodine, but it was just sitting on that layer there, and it couldn't get fixed to peptidoglycan. So the alcohol washes that away. So by this stage now, you've got bacteria that are purple coloured and bacteria that don't have a stain. So that you can see them all under the microscope and differentiate, you then add saffron in. So saffron in is a pink stain, which will then bind. So that binds on afterwards. And then down the microscope, you can identify purple bacteria, which are gram-positive, and pink bacteria, which are gram-negative. And that's because the purple ones have the crystal violet stain and the pink ones have the saffron in, the gram-negative. And it's all because of that lipopolysaccharide layer. If you're identifying some bacteria, you could do that stain technique first of all, and then that gives you those two big broad groups, like I said before, and then you'd have the stain on to look and see the shapes, and then do other testing to see what bacteria you had. Okay, so bacteria can be grown on plates, and it's grown for lots of different reasons. So one of the reasons is if they wanted to do a hygiene check of something like a hospital surface or in a restaurant or something like that, they could swab and check for hygiene tests, and then grow what's there onto an agar plate and so they can see it. It could be that someone's got an ear infection that won't go away, something like that, and they might swab that to see exactly what bacteria it is, so they know what antibiotic to give. A lot of the time, though, it's to do with testing products like antibiotics or testing antibacterial sprays, wipes, household items, cleaning items, all that sort of thing. So they would need to grow lots of different types of bacteria so that they can say, like on the adverts, it's 99.9% of bacteria are killed, all that sort of thing. Grow the bacteria first so they can test it. Also testing the strength. You don't want to use a cleaner that's too strong because it's not safe to use in the home or something like that. So testing for the strength and for what bacteria are killed by something, as well as checking for what type of bacteria people have as infections or for swabbing and that sort of thing. And the way that it's grown is to grow it in the lab, and you may have done this in year 11, if you remember, using aseptic techniques. Aseptic technique is the one where you have maybe some alcohol and a flame, a Bunsen flame, and with your inoculating loop, the one that you're going to spread the bacteria with, you would have to, it's dead awkward if you're not used to it, you'd have to pass the bottleneck, so a glass bottleneck, through the flame to kill bacteria around the bottleneck. You'd have to pass your inoculating loop through the flame. Don't put anything down. Open up your Petri dish just a tiny bit like this, because you want to let bacteria in from the air. So just open it up very slightly, and then use your inoculating loop to dip in the bottle and then spread on the plate. Okay, and then close it up again and store it at the correct temperature, which I'll come back to. And the way people spread it on plates, there's different ways. One way is to actually mix in with the agar. Another way is to have the agar set and then drop it on and swirl it around so that spreads it completely. You can also get a spreader that you might have used before. Your teacher will have said, don't melt it because these are plastic, we have plastic ones. It's like an L-shaped spreader that you can really try and spread it all over the top surface. And that would be a lawn spread where it's completely, like a lawn, completely covered. Another way of doing it is to have your inoculating loop and do like a little spread, and then without dipping it again, spread again and spread again, like that sort of shape. And the idea is that by here, the colonies will be separating out and you'll be able to count them and look at them, look at the colours and things like that. Maybe not count like that, but you'll be able to look at them. Another way is to get a big squiggle, and hopefully again by here, you see the colonies separate out so you can look at them. And like I said before, the lawn spread. You can then sellotape them, if you remember, like a north, south, east, west. Because if you tape them all the way around, you grow anaerobic bacteria because no oxygen. So they're often dangerous. Well, they can be. So we don't do that. Not normally the type of bacteria we're trying to grow. Another thing about storing them, once you've done that, you need to store them at the correct temperature to let them grow. So in school and in lots of places that aren't sort of heavily controlled labs, it would be around 25 degrees room temperature. That gives quick enough growth. If it was 5 degrees, it wouldn't grow fast enough, it wouldn't be enough growth. 25 degrees gives quick enough growth, but it's not near to 37 because that is body temperature. So obviously that would be pathogenic bacteria that you'd be growing there. So ones that survive well in the body. So that's only done in controlled labs. Then afterwards, you want to kill it off. It's 121 degrees minimum, and sometimes done sort of under pressure as well. But you get rid of the bacteria at 121 and above degrees, or they could be irradiated as well. So bacteria is grown for lots of reasons, and in those ways. The way that you need to learn about is a serial dilution, and that allows you to do a viable count. So a viable count is where you have just counting the live bacteria. So what you would do for a serial dilution is to have, say, 1 centimetre cubed, 1 milliliter of your bacterial sample, and then 9 millilitres of distilled water. Distill it to get rid of anything else that might be in there. And then that gives you a mixture that's obviously 1 in 10, the bacterial solution. So 1 in 10, or 10 to the minus 1. Then the next one, you take your milliliter this time from there and put it in there. So you've already got it at 1 in 10 dilution, and then that 1 milliliter again goes in with 9 millilitres of distilled water. So now you've got it 1 in 100. Okay, so that would be 10 to the minus 2. And you just keep doing that all the way along, and you will do this at some point when we're back in school. So 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 4. So you get to the point it's 1 in 100, 1 in 1,000, 1 in 10,000, 1 in 100,000. And you grow the bacteria from all of those dilutions because we want to see which one is the best for counting for our bacteria. This plate here will have so many colonies usually that it's difficult to count. They might all be merging and difficult to see, and you just lose count. So down here it's not going to be accurate. Okay. As you go further this way, the accuracy will improve. You'll be able to separate them out and count them, but there still might be too many. Down this end, so too many here, like this. Loads and loads really, really, really close together. Obviously less in there, but still too many to count. Less. And less. Then when you get down this end, it becomes unreliable because there's not enough to give a decent sample. So you'd have a low sample here, and it wouldn't give you a good result. So what you actually need is one that is between 25 and 250 colonies. Okay. What a colony is where one bacterial cell, a single living bacterial cell, has landed on that nutrient agar, grown and divided, so that it's made its own little group of the products of its divisions, basically. So like it's got this little family with it, and it grows enough and divides enough that you can physically see the bacteria, and that's only because there's so many all in one little clump. They're like little shiny blobs. So you'll do this. So when you get these colonies, we're assuming that each one represents that one living cell that came out of that solution into there. You would have got your spreader or your loop, put it on, or just swirl it around, put it on, and then left it to grow. And from these different dilutions, you'd select the plate that's between 25 and 250. And it's a reasonable amount, count them up, and that should give you a viable count. So it should tell you how many bacteria were living in that sample. If it was this one, from 1 in 1,000, obviously have to multiply it back up by 1,000 to get your results. So you'd have to know which one you used, which one you selected, and then multiply it back up to see how many bacteria are in the sample. The nutrient agar, this is one of your questions that a couple of you struggled with. The nutrient agar has to contain the carbon that they need, the nitrogen that they need, growth factors, vitamins and minerals, and the correct pH. Water is not really, because it would have to be the correct amount of water to make the jelly the right consistency. Oxygen, you'd give oxygen, it should just be the right, oxygen in the room, so that's not correct really either. And it certainly wouldn't go into the jelly in that way, or be put into the jelly. And then temperature, again, is a factor, like I said before, but it's not in the jelly. So there was one question there that some of you struggled with, so it's got to be something that's in the jelly. Carbon, nitrogen, growth factors, vitamins and minerals, and the correct pH. So you'll get a count from that. That's the viable count. The total count is where you can count the living and dead bacteria. You don't need to be able to explain how that's done. It's basically, you can do a serial dilution and count under a microscope, under a strong microscope, count the cells by looking at them physically down a strong microscope. But that would be, again, in a diluted sample, and then you'd have to multiply it up by what you see in that view of the microscope. Okay. The other thing I mentioned is the bacteria that grow in these plates. They might be different types of bacteria. Obligate aerobes are ones that have to have oxygen. Obligate anaerobes are ones that have to be without oxygen. And facultative anaerobes are bacteria that can use oxygen but can also survive for periods in anaerobic conditions. Okay, and that is the main points of the topic really there summarised.